The objective of this event is to exchange experience feedback when it comes to measuring biodiversity near a seaweed farm. These researches are still recent with a few perspective on lead experiments. That is why we are seeking for more collaboration. We also need your feedback while preparing a new study here in France to measure biodiversity near a sugar kelp farm, which Melanie on the AlgoESCO team will provide more feedback on in a few minutes. So thank you for your attention.
Thank you, Philippe, for this introduction, and thank you all for joining. I see a lot of interest. It's almost 80 people attending, and always we are publishing the workshop online. So yeah, very delighted to be able to gather such a group of experts because we have in the room marine ecologists, biodiversity experts. We have the Scientific Council of the Global Seaweed Coalition, which is spread around the globe. We have institutions such as FAO, WWF, funders of projects of biodiversity, and also seaweed farmers. So I think we are a big group, and I'm looking forward for the discussion. So I will guide you and share my screen now on what would be the agenda and the purpose of this workshop. So yeah, just a few words about the science and technology pillar, even though Philippe introduced it well. So we gather 15 independent academic researchers, and that's important for the coalition because they give us perspective and support when we organize call for proposal to give their objective perspective on different topics related to seaweed. We lead a workshop. So the last one, as mentioned, was on biobanking, and we developed a nice collaboration since then with FAO, trying to deliver on an Aquapolis platform that was just launched. We'll be present next year in a conference such as ISS, International Seaweed Symposium in 2025, and the One Ocean Science Congress in Nice, France, happening before UNOC. We also support the launch of a seaweed breakthrough, which is led by the Global Seaweed Superstar team at the National History Museum and SAMS, and one person will be presenting today at Nice. So we funded different projects, more than 32 projects since the beginning of the coalition. And some of those projects are related to measuring marine biodiversity or abiotic element near seaweed farms. So there is Kelp Forest Foundation that will present today a baseline of what they have run as eDNA study. Then there is Nocibora seaweed, which has been measuring abiotic parameters near a seaweed farm, and that is now inviting a research project as a pilot farm linked to seaweed farming and coral restoration. And last is the project awarded this year in AlgoESCO, which I will present a bit more detail now before the AlgoESCO team come in presenting. So the AlgoESCO is a commercial seaweed farm. It's based in the northwest of France. And so it's a brown seaweed farm. They have Saccharina latissima, Undaria pinnatifida, and Alaria esculenta. It's a farm of 150 hectares, producing 200 tons of dry seaweed. So if I'm not wrong, it's the second largest in Europe in terms of volume, following ocean rainforest. So you have a picture of the site on the right. And so the situation at AlgoESCO is that they've been asked to provide impact study near the seaweed farm, and they've been done, they initiate studies 11 years ago, and each year they have to produce a report. It costs the farm 30,000 euros to provide those reports. So it's an inventorying of rocky substrate and soft substrate, which is sand under AlgoESCO farm, and they run a study of benthic fauna and flora. Yet there is no measurable impact of the seaweed farm on the marine ecosystem, which is good because there is no negative impact, but we think it's worth investigating a bit more, and that's why the project came out of trying to combine a protocol with using environmental DNA, arms technology, underwater camera to try to find something around this farm. And that is why we organized initially this workshop to get experience feedback of all people that have run biodiversity study like that, to get your experience feedback and to build a really strong protocol. And that could lead, of course, to further collaboration later. So we have a tool here in Roscoff, so that's where Philippe and I are based. It's called the Genomic Observatory, where we have researcher that's been studying microscopic plankton and the impact on marine biodiversity through machine learning, arms, etc. But we still need, it's still interesting to get your feedback to use at best those tools. And so lastly, before I give the floor to Algolesco to present what they've been doing in terms of study, here is the agenda for today. So we'll start with 4 presentations. So Algolesco, Sophie Kor will be presenting a work within the CIMAR project in the Feroe. There will be Kelp Forest Foundation, as they've done an eDNA baseline. They will share their results. And we'll stay in Africa then for a project led by Isabel Obina Barreto that's cultivating near the seaweed farm and about reef restoration activities. Then we'll have a Q&A around those presentations. If we have time, we'll do a break, and then we'll move to a second round of presentation, because I want you to be awake, because there are very interesting speakers today. So there will be Jose Maria that will present his work about the project Genial and the impact of seaweed farming on seagrass. Then Anne Brun from Denmark, she had some interesting results to share because they did measure upstream and downstream of 2 seaweed farms and through eDNA sampling. Then Sophie Corrigan, she has run an interesting work on trying to standardize protocol for biodiversity check. That was 3 years ago, and the technology has very much evolved since then. So we'll see how we can take this work and maybe update it. And lastly, we'll go to Canada, and I know it's very early in your side of the world, Cascadia, but I think it's very interesting they developed underwater camera and an algorithm associated to it to be able to read more than 9 hours of movie to count the fish near the sugar kelp farm. And then we'll open the floor to some questions. So I have listed questions. So what are the best approach to maximize the use of eDNA? What are the most cost-effective solutions to measure marine biodiversity? Would you be able to share your protocols as you have led some study with us? And how we can leverage those results for biodiversity credits? So with that, I'm very excited to welcome Corentin Marty and Mathilde Moine from Algolesco to introduce their work. Over to you.
Hi, everybody. So we are going to present you our environmental monitoring that we are doing currently in Algolesco. Next slide, please. Okay. So Algolesco was created in 2013, and we exploit a marine territory of a substantially side with 2 main areas where we produce kombu royal. We can go to the next slide. Thank you. So we produce mainly on l'Esconil and Moëlan-sur-Mer with a total of 340 hectares. We also produce wakame in a smaller farm. So Algolesco In 2090, Aller Company joined Algolesco with a 30-hectare farm in the north of Brittany. But today we are going to talk about Leskoning farm. It's about 2 miles away from the coast with a surface of 150 hectares on a deep between 15 and 20 meters. It's an open sea farm exposed to winter storm. The maximum current is around 2 knots, and it's mainly sand and rocky bed.
Okay. Well, next slide, please. Yes. So yeah, hello everyone. So when the project was set up, the French authorities asked us to propose an environment monitoring protocol. So first we will look at the monitoring on rocky surface, following by image of the structure, and the studying will carry out on the soft substrate. Next slide. Well, next slide. Quickly, when we look at the various publication on impact, we find some positive and negative point around sea farming. The idea was to think about which point has no impact and which has an impact that might be relevant. By studying all these points, we focus our research on 2 aspects: the influence of cultivation on the algae population around the concession, and the enrichment of the sediment in organic matter under our marine concession. Next. So for the rocky monitoring, we have made a maximum use of our equipment available, but we call professional divers with biological training. 2 points are studied, one outside the concession and another one outside. The idea was to use, next. So the idea was to use a monitoring protocol set up by Ifremer following major work on the Brittany coast. But after a few years, we had to reduce the number of studying points because of the very high cost. The number of dives was becoming too heavy to support. For now, we can understand that kombu cannot grow under our algae field because of the deep and the turbidity.
Just need
This, no, sir. But this protocol is interesting to look at the state of the habitats and identify the different species present. But in the other hand, it's difficult to understand the fundamental question of the influence of algae farming on the surrounding environment. But the idea is to continue diving under and outside the concession for the moment in order to continue assessing the states of the rocky habitats. Next. The real challenge of this protocol is to cover the significant costs associated with the divers because we have to find the knowledge, skills, and equipment, and that's we don't have as a seaweed farmer. Alors, placed on the seabed, our concrete blocks that you see are a good source of information regarding to the colonization of marine species. On the left, you can see buoy covered by mussels. And at the right, there are photos of our hook, more or less colonized by other small algae. It's depending of the deep where they are located. It's in reality, these photos is only for observation, but it can be an interesting idea to base a scientific protocol on it. Next. So as I say, we looked at different positive and negative points to purchase our monitoring. So therefore, we focus our efforts on monitoring marine sediments, the seabed population and their habitats. So first we had a look at the sediment composition. Next, we had to look at the evolution of organic matter and understand where it comes from. In order to understand the question of the origin of organic matter. Next, the composition of benthic communities in the sediments. And over the years, we added the analysis in order to understand the change and their impact. So as nitrogen, phosphorus elements in the sediment, chlorophyll A and phytopigment in the water. So all monitoring have been carrying out at all time in and out the marine field. Next. Alors, for the sediment study, it may seem simple, but it gave lots of things to think about. It. In 2029, we have been already cultivating for 6 years, but in 2029 we could no longer find silt in the sediments. So why was it? So we've been hit by a major storm during the winter time, so which obviously right, moved the sediments underneath the concession as well as outside of them. So it's an important concept because it reminds us that the seabed is not static, and below the sea many things move, and we are not able to see them. Okay, next. Ah oui, alors ça c'est so this disposal of the sediment clearly showed that the different effects on the total organic matter, and we can see that the bottom of the concession and the control site follow relatively the same curve. It's also interesting to note that the TUOC can change over years. Next. Alors, quickly, the same applies to nitrogen and phosphorus concentration, where the impact of seaweed cultivation is not really visible. Again, 2 studing spots have quite same movement. Right, next. So we therefore wanted to understand if the carbon in the sediments is coming from our algae. So we try to use the stable carbonate isotope, stable carbonate isotope to compare. So this analysis is a direct answer to the question of impact. We had assumed that growing algae could enrich the environment with organic matter, but we have now what can prove that the substrates are not in fact made up of carbon from our kombu residues. Next. To finish these studies, we try to identify the population present in the sediment as the rebent studying and monitoring in France. So here we have understood that the density and richness of benthic population is very close to the years of sampling. So the impact of seaweed cultivation on their species is very difficult to check. Next. Well, using all those data, we obtain ecological status. So 2 interesting indicators are used to estimate the balance of population. So it's Shannon and the sensibility of species to disturbance, so AMBI. So once again, we had noticed that there are changes over time, and 2 monitoring spots follow the same changes. So we wait for the 2024 results, so this year's, because Kiaran, the storm, may once again cause movement on the seabed, and it will provide us some very interesting data. Right, next. So for the conclusion, what is important is the strong influence of marine environment on the sediments. Chemical data in substance evolve in the same way between under and outside the marine concession, and the same ecological status evolve over the time. And to conclude, is all the time it's difficult to prove that there's a really interaction between our alga field and all the biodiversity and marine biodiversity. Voilà. Thank you for your attention.
Thank you so much, Corentin, for sharing your data, and I'm sure you are not the only one in this situation. So we'll hear from more kelp farm soon, and I hope we'll get inspiration from the next presenters. So with that, I'm welcoming Sophie Kor, I hope I pronounce well your last name, to introduce a work within the CMARC project based in the Faroe.
Yes, thanks so much. So yeah, the CMARC project is a Horizon Europe project, and we were part of Work Package 9, who was doing all kinds of ecosystem service assessment, and part of this is the biodiversity assessment. And we did that on the ocean rainforest farm. We also have ocean rainforest present today. So in a big team, we did a bunch of data collection, which I want to present today also. Yeah, next slide, please. Yeah, basically, I'm going to talk you through where we sampled. So what's the context of the Faroe Islands, the ecological context, and what we sampled, how the experimental setup was. And then a little bit in detail on the manual and the eDNA samples that we did, which led us then, or like in the presentation, which leads me to the research question that we think we can answer with this data. And also then a tiny self-evaluation of where is our data collection strong, but where is it not so strong? What data are we missing? And this is where I think this workshop is super interesting, then because I will, you know, maybe get some input on what else we can do as a follow-up. And yeah, and I end with a few reflections. You'll see, I'll probably rely on your presentations later on to answer them. Okay, next. Yeah, so basically, where are the Faroe Islands? They're in the middle of the North Atlantic Ocean. And next, yeah, which leaves them to have a very stable water temperature between 5 and 11 degrees, which is brilliant for seaweed aquaculture, as you can imagine, because it doesn't get too warm. So you can even harvest multiple times. And ocean rainforest is harvesting, I think, up to 6 times within 3 years. So they harvest partially and they just leave the structure in the water, which is also good for biodiversity. We can test what it means over the years to analyze the structure. And as they are within the Gulf Stream, they are pretty exposed actually to currents and waves and weather and everything there is. But as you can see here on the picture, it's still very near shore. So this is one of the few sites that are actually considered exposed. Next, please. Yeah, basically saying the same thing again, that the cultivation structure stays in the water for longer. And also due to those nice currents and waves, they have a really good input of nutrients, which makes them able to grow in a 3-dimensional structure, which means it's not only the long lines, but they also grow vertical. So there is 10-meter long lines vertically. And due to the currents, actually, they even down there still have up to 2 or 3 meter long saccharina. So it's really, I think it's not so often, or even one of the few or the only one that actually is able to do that right now. Next, please. Yeah, so the experimental setup, what did we do? So we sampled in July and in April, which is sort of the beginning of the growing season, or like mid of the first growing season of the year. And then July is like full on second cycle of the growing season. So what does that mean for biodiversity? We also compared 2 of their sites. So one site is like quite out in the fjord and the other one is quite into the fjord. So what does that mean? With different currents and other different species settling and so on. And then as they have their cultivation lines for 3 years in the water, even we sampled on 1st-year lines, 2nd-year lines, and 3rd-year lines, and hope that we can, or we will compare those lines. And their lines are up to 10 meters low in the water. So we sampled on 2 different depths, which is also a nice comparison to see, is there different species down there than up there? And in our method of assessing biodiversity, we also checked, we wanted to have something really quantitative. So we manually sampled and we complemented that with eDNA. And as you know as well, eDNA doesn't necessarily give such good results on quantitative measures, but for sure it can validate, for instance, our biomass that we sampled, because we sent that in for eDNA as well. So we want to validate if the species that we identified are really the ones that the genomes also say. And it also is a complementary data because we took water samples. On the next slide, please. because this water samples, we might be able to capture different species than with the biomass. So we sampled within the farm, so where the first cross is, and then at the edge of the farm, and then 500 meters away. So that is also like a gradual distance to the farm. Is the biodiversity richness or the species going to be different outside of the farm than inside of the farm? And also on natural kelp within the same fjord and also in a different fjord. You can click next. There is a couple more crosses coming. Yeah, exactly. So so that we have a gradual a difference between the biodiversity of the eDNA in the water in the farm and outside of the farm, and also on the natural kelp. Next. Yeah, so just a few pictures on how the manual sampling went. So we designed a 2 meters in diameter net, which is like on a metal ring. And because we had to lower it down to lift up a whole vertical line. So it's a 10-meter long line with it was up to 50 kilos heavy, of course, including the line, but of biomass on it, of seaweed really growing on it. So we lifted up into the boat with a huge crane, and then took samples of the upper part of the rope, of the lower part of the rope, put that into sample bags, and then also rinsed off the whole 10-meter vertical line to capture all of the mobile species. So here on the left-hand picture is actually already, then we obviously cleaned it off, which animals are in there. We put them aside, put them in ethanol, counted them, identified them. And here you see actually a mobile species. The M stands for mobile. So this is how it ended up here in Kiel, where I'm sitting today, which was then treated for the eDNA. Next, please. Yeah, so eDNA, also a bunch of pictures, because it was quite challenging actually to capture or to get the water. We didn't have the fancy tools where you lift something down. We actually had to send a person down. We couldn't reach it just from the boat by leaning over, because you don't have, you're not supposed to break the water surface with your water, with your bottle. So we actually had to send somebody down and hold them tight with a rope. It was quite cold, but yeah, it was also fun, I think, more for us than for him. Next, please. Yeah, the rest of the eDNA, you know it, it's not as interesting anymore, but obviously we filtered it, and now it's here in Kiel in the lab, and it gets smaller and smaller and smaller, and extraction is done, amplification will be done, and sequencing within the next few weeks. Next, please. So the research question we're trying to answer with this is, obviously, first of all, the cultivation unit of the Ocean Rainforest Company, what kind of biodiversity do we find within there? And also we can say quantitatively how many species are going to be there. Obviously, we're going to extrapolate the data that we have on the rope samples, and then say something about the whole, per kilogram of the whole harvest, basically, what there was. What species and how many of them there was within this one or the several cultivation units that we assessed. And since we have eDNA water from outside of the farm and from natural kelp, we also want to say something about how does it differ, the biodiversity richness and the evenness in the farm towards something outside of it. And I think there's one more question, if you click, yeah. And then we have these differences within the cultivation unit that are quite nice. So the different exposures, because we're comparing the 2 different sites. And also we have these 3 different years. So we have 3 replicates of years of each line. No, 3 replicate lines of each year line. And then also the seasons, because we have the data from April versus the data from July. So there we see already, like with our own eyes, we saw the difference. So the data is going to hopefully show the same thing. Next, please. Yeah, so I think the strengths of our method are that we are combining the manual method where we really have quantitative data. We can say we counted it ourselves. We had our nose in the ethanol, and we really know that these amount of species are in there. And also coupled with the eDNA to confirm it, to complement it as well, and to have that gradual distance. How is it further and further away from the farm? And we can make all of these comparisons. However, there are weaknesses because we only have a few samples from outside of our farm. There are collaborations or discussions going on with another team that actually did more samples in the natural kelp. So we would like to share and compare these data, and that would be absolutely brilliant because they're sampled in the same fjord, so that would be nice. And but we weren't able to take more data ourselves. And the other weakness, I think, is that we weren't able to capture so many of the fish and the mammals because obviously with our boat and the crane and the net, they would just swim away. We did catch some fish, smaller fish, but there is a lot more there that we just didn't catch in our net. So we're missing that. And that's actually interesting or relevant for our project, for the context of the project, because as a follow-up of this task, we're supposed to value it monetarily. So obviously something like commercially interesting fish would have been interesting to have, which we weren't able to catch. But we're also planning on a follow-up project where we might want to use some cameras and so on. Next, please. Yeah, so my reflections basically are, and these go really into the discussion as well, and I'm very much looking forward to presentations of all of you. So we really tried and reflected a lot on how to best quantify fish. I mean, are you actually going to you cannot count every fish there is in a fjord. So what technique can you use to make a valid and proper scientifically valuable assessment on the quantity of the fish? They're so mobile, and how can you say that they're not just coming from the natural kelp towards your farm? How can you say no, they're here thanks to your farm? And so I know of these techniques of baited cameras, but they could also come from all over the place. And are they really additional? And I would be interested in, for example, has anybody tested something on little juveniles that that hide in the seaweed? Because what about survival rates of juvenile? Would that be something to quantify the more fish that is there thanks to the cultivation unit? And also a different question that we encountered in our project is that right now we have to value this biodiversity. And I think every assessment always has, like a different context to it. So are you doing this biodiversity assessment because you do have to do a monetary evaluation of the biodiversity later on? Are you doing it, I think, as I understood more like the Algolesco case, are you doing it to show, no, there was no harm done here right now. This is either, beneficial, but we're certainly not doing any harm. Or are you trying to say, no, we want to, you can click a couple more. Yeah, we actually want to restore some habitat. So are we trying to prove that, one back. We're trying to prove that basically the seaweed cultivation is just the same habitat as the natural kelp. And this question again with the attraction or the addition, are we having additional more biodiversity, or is it just moved and has more space to hide? And for instance, another thing would be interesting is, are we finding any invasive species? Obviously, that would be then something interesting to show, or even more habitat for endangered species, or is that even a problem if you have endangered species? Because eventually your structure is going to come out, or a boat is going to come by. So yeah, these are just reflections that biodiversity assessments can, I think, have very different goals, and and what are the best techniques for each question. And I hope we can answer some of these questions in the presentations or in the discussions. And that was the end of my presentation. Thanks so much.
Thank you, Sophie. Thank you so much for sharing those relevant questions, I think. And just for us to know, when is the CMAR project ending? When can we expect the result of your study?
The result of this study is actually going to come quite soon, because in February we have to hand in the deliverable, and there also is going to be a publication about it, and that's going to, maybe mid next year. But in February is going to be an internal review, at least, of the deliverable, and I don't know at what time that is going to be out publicly. But Wood is here in the call; she might know that. But sometime next year.
Thank you very much. And so with that, we're going to leave Europe and go to the African continent with first study based in Namibia, and then to travel to Madagascar. So Kelp Forest Foundation, over to you.
Thank you. Good day. On behalf of the entire team at Kelp and the Kelp Forest Foundation, I'm very honored to provide this presentation where I'll talk about the 2-year biodiversity baseline that we did here in Namibia. Next, please. So the establishment of the biodiversity baseline was very essential for us to lay that foundation, and that can usually be used to then track impacts after the farm has been established. During this biodiversity baseline, we did it by providing scholarships to Namibian master students. I myself was a scholarship doing it. I did the algal baseline where my counterpart did the fauna baseline for it. And we used different techniques, different sampling techniques, from spotting to quadrats to phytoplankton using the net. And then with the Global Seaweed Coalition supported us by using eDNA technology. And we're one of the very few in Namibia to have used this technology to monitor biodiversity. We monitored at the Kelp Blue cultivation farm, or their potential cultivation farms. And this is a privately owned company that cultivates with giant kelp. And I'll continue to talk more on that and the process. And we work very closely with Naturmetrics, who is the global nature intelligence technology providing us natural monitoring. They provided us with the eDNA kits, which is super easy to use. And they also then extracted the DNA and then provided us with different reports. Next, please. Thank you. So at Kelp Blue, where we did the biodiversity baseline prior to the establishment of the kelp farm, we have an integrated value chain, and we are a part of every process, so starting from the onshore cultivation. We have a hatchery right here where we get our spores, inoculate them, and then induce them. After that, they get taken out into the water, where we will then seed them on lines. That's below the surface. And then we have a very dedicated marine monitoring team that goes out and monitor the different changes that we have, or that the impact that the environment has on the kelp as well. After our kelp reaches the canopy or the surface of the water, we harvest only the canopy. So we only harvest the top bit, and then the rest of it is left behind for the main aim of Kelp Blue, which is to enhance biodiversity and store away carbon dioxide. After we've harvested, we take it to our factory where we use everything. We do not leave anything for waste. And the first product that we have at Kelp Blue is our organic fertilizer, Stem Blue, and then that goes on to sales and distribution. The rest that stays behind, so the pulp that stays behind essentially, after we've extracted all the liquid bits for our organic fertilizer, that goes into R&D, where we play with it, where our team plays with it to come up with different products such as kelp leather and kelp foam. So that's just a nutshell in Kelp Blue. But we're here for eDNA, so the marine monitoring team, which is the dedicated monitoring team that did all this baseline, used environmental DNA. So environmental DNA is so important and can be used for such diverse different things. So conservation biology is one of them, and this is through detection of, endangered species or invasive species. Ecological research, this is looking at the functional diversity, the community and ecosystem processes, and what we've used it for, which is biodiversity monitoring, to detect species and to look at the whole community composition. It can also be used in fisheries management, which is something that I think we can adopt here at in Namibia, to look at to assess our stocks. Next, please. Now I'm going to go into the methodology. So this was a 2-year baseline assessment. It started in December 2021. We did sampling every single month at 4 different sites offshore, and then we had 1 control site. The primers that we used or that we analyzed for was vertebrates and then eukaryotes that encompasses everything. This was done in the area where the kelp farm would have been, and then the surrounding areas. And this would have been upstream and downstream. Kelp Blue also has 2 farms. We have a smaller inshore farm, and then we have a bigger offshore farm, which is just north, if you look at the map, so just north of Lutritz. Next, please. So we had sampling kits that was sent from Naturmetrics all the way here, and it has everything in it that you would require to sample and filter the water. We then start with obviously preventing contamination because it's extremely sensitive, and it's also one of our shortcomings with this particular method is it literally picks up animals that are not supposed to be in the ocean. But we have a really good communication and relationship with Nature-Metrics. So we always went back and forth, and we said that, you can't get, a gemsbok chilling in the ocean or, a kudu. It just doesn't make sense. But we prevent contamination, and after that, we collect water from the ocean using a clean bucket. And then we pump it through the filter using the vampire sampler. It's in the name. So basically what it does is it sucks the water and it pumps it through the filter, makes it super fast instead of doing it by hand. And then after we then preserve our filter that has all the eDNA trapped in it. And this can be stored for about 2 weeks and then get sent off to Nature-Metrics, where we've completed the whole data sheet that comes within our sampling kit. And then they do the rest where they then extract the DNA, amplify it, and then match it with their database. Next, please. For the results, next, we have the number of distinct species over the years or over the 2-year process or 2-year project. You can see that it is quite distinctive, and it does pick up over the quarters of 2023. The places where we don't have or you don't see any bars is essentially we're still waiting on some reports to come back from Nature-Metrics. And can you still hear me?
Yes.
Good stuff. And then sometimes we also take samples and then we send it, and then it sometimes doesn't get picked up properly, so we don't get all the results from it. That's also a possibility. It's also one of the shortcomings. And that's something that we're working on by doing replicates from here on out. Next, please. This is also just shows the cumulative number of distinct species. There's all sites. These are all the sites combined. And then shearwater bay is what we call our smaller inshore kelp farm, and it will show you the difference within the kelp farm and then from the kelp farm or outside and around the kelp farm all over the years or the months throughout the years. And you can see that it picks up significantly for both of the sites. And then we reach a point where it kind of just levels out. And this essentially just means that I think we've reached the monitoring capacity of eDNA. So it does happen, but it has helped us a lot. And if you go to the next slide, you'll see the amount of different species that we get from just filtering 4 liters of water. That takes about 10 minutes. We get a huge variety. We get, from our tiny phytoplankton all the way to our big animals, all the way to our dolphins and birds. And then what we basically do with this is by using the normal method of sampling, and then this will then complement it. And then we do water quality. So we look at nutrients of the water as well, just to see if that also has an impact. So we just can't say that because our kelp farm is there, we've seen an increase or a decrease in biodiversity. So we're looking at all aspects to explain the changes better and to lay that baseline. Next, please. There's obviously a lot of limitations and advantages. The limitations that we have is it doesn't give us any age or size data. There's no actual counts. Unfortunately, we don't know if it comes from one specific individual or it came from a group of individuals. Sometimes there's an incomplete reference database where we only get certain species that are identified to a certain class and not further. That's also sometimes tricky. And then the spatial uncertainty because of the size and the movement in our oceans. We don't really know if it was at that specific area at that time. And then the lack of standardization. But regardless of that, we have a lot of advantages. I mean, it gives us a big variety, a big, large data set for monitoring. This also reduces the cost that we go out. This reduces the health and safety risks that, usually is accompanied with being in a field and collecting data. It detects cryptic or elusive species. it works in all types of water as well as different sediments. So some of our other master students have also used it to look at sediment, eDNA in sediment. And it can actually be used by anyone. It's a non-invasive method of sampling. And so we're continuing to do this even after we've established our 2-year baseline. We are going to kick off another project soon where we are doing it every quarter now. This is to look at the impact that we're going to have, or the impact that we potentially could have. And we're also changing it from 1 sample per site to about 4 samples per site to have replicates as well as a control. Yeah, but this is about it, and we're excited. And we thank the Global Seaweed Coalition for this opportunity. Thank you.
Thank you so much, Angelique. And yeah, interesting to see that you highlight the same limits of quantitative result linked to eDNA. Also interesting that you have raised all this amount of species, meaning you have all the primers to be able to analyze those species out of your samples. So that's a strong budget. And the last point I wanted to raise would be interesting if some of you have done eDNA sampling in sediments. From what I understood, for Care Forest Foundation and for CMARS, it was from seawater. And I think sediment is an interesting point to look at.
Definitely.
So we'll have one last presentation before we go in the first round of discussion. So Isabelle, actually, are you going to share your screen? So I will stop sharing and I leave it to you to share your study in Madagascar.
Yes, thank you so much. So here we go. Normally you will see now my screen.
Yeah.
I put it in slideshow. Excuse me. I am currently in Madagascar, so maybe my connection is not so stable.
It's okay, otherwise we can see your slides, so you can go like that.
Okay. Okay, if it's in now, it's okay in full stream?
Now to use the option.
Okay, no, it's okay. I try this. Maybe now it will be okay. Now it's okay?
Yeah.
Okay, so here we go. So thank you so much, Melanie and Philippe, to invite me to share our work in Madagascar. Our title today is Possible Benefits and Impact of Tropical Seaweed Farming and Reef Restoration Activities, a potential for natural-based climate solutions in Madagascar, Western Indian Ocean. Is my postdoctoral project that I started by the end of 2023, and I will work for this project for 1 year more, and I hope that we can obtain some funding extra to continue with this fascinating domain of seaweed farming. So let's begin with some context. If my presentation wants to work. Okay. So tropical coral reef ecosystems are one of the most biodiverse in the planet. They host more than 25% of marine biodiversity. They represent an economic value that more than 500 million people depends for their goods and service. And unfortunately, more than 15% of this reef habitat are already lost. And from this constant, international organization support different action to preserve socio-ecosystem that will determine socially just sustainable development and biodiversity conservation, mainly in the developing countries. So nature-based climate solution and their potential in the mitigation of greenhouse gas are investigating worldwide. And seaweed farming is a relevant activity that can represent a high potential to this mitigation of caused by the climate change. And also seaweed farming represents a diversification of the blue sector to ensure incomes in different societies. Restoration solutions are needed now in a world that different ecosystems are in erosion, in the erosion of their biodiversity. And from this concept, our research wondering about the benefits of the co-culture of corals and seaweed. And we try to correspond to different sustainable development goals. So tropical seaweed farming and reef ecosystems, this activity represents a high economic impact in the development of the blue economy. In Madagascar, this activity beginning by the end of the 80s in the southwest of Madagascar, using a red algae, and from the last 10 years, the activity was increased and the national production are accelerated. The principal algae produced is Kappa ficus for the Caribbean, and the regional platform represent today more than 1,600 seaweed villagers. And the main seaweed production companies are ocean farmers that are located mainly in the southwest of Madagascar, and Sovalg and Nocivora seaweed that are located in Saint Mary Island in the northeast. So why the Western Indian Ocean and Madagascar? Because Madagascar is one of the most important hotspots of biodiversity, and this country, amazing country, plays a key role in the conservation of the regional plants. Madagascar represents a high potential to develop blue economics, notably in the coast activities and the seaweed farming activities from the last 50 years, complementing fisheries activities that are in rarefaction for the last 2 decades. So IRD, the French Institute for Research and Development, mainly take action to support the sustainability of different actions and activities like seaweed farming. Is the reason why we choose to work in Madagascar, and I start also with the acknowledges for the collaborators. We work with different organizations, societies, but also national organizations for investigation in the halieutic domain of research, also for the ONGs or communal and local organizations. And we work with different laboratories. So here today I will present the integrated approach, the study sites, the ecological. I will do a focus for ecological issues and give some preliminary results. So Resto Ecodurable is a sustainable science project, and we main research and interdisciplinary approach, and we develop 3 axes: ecology, society, and law. And today I will present more the ecological study, because we are in a biodiversity workshop. So I will start just to give some elements about our study site. After some exploratory field, we choose to work in Sainte Marie, specific in Nocivora seaweed farms. So we choose this site that the activity start from the last 3 years, and but the activity in Sainte Marie beginning 7 years ago. We choose also this site because in Sainte Marie there are a creation of a marine protected area, and this is a current project, and the application of this marine area is a current action. So we find socio-economic and environmental issues very interesting for our research. So here we try to do in the ecological axis sorry, I lost my screen. To describe the habitats and the biodiversity in the Reef Lagoon, to describe physico-chemical parameters in the different sites of farms, of seaweed farms. And we will start an experience of co-culturing corals and seaweed. So for physico-chemical parameters, we will start to analyze some Nocivora seaweed data, and we will take some data with our actual field campaign. So different parameters: temperature, salinity, nutrients, chlorophyll, and some extra parameters for the experience with co-cultivation, algae, and corals. So as I say, I will focus today in the ecology axis. So I will present some results for the current description, also about the monitoring of different physico-chemical parameters. Some results about the line intersect protocol that we aim to describe Benti communities, and some census of biodiversity, and some results also from drone surveys that are very complementary data for the description of all the study sites and different hypotheses that we want to explain in our research. Environmental law and anthropology research are very important, and at the same title of ecology, but I can take some question after the workshop, or we can contact me for more details about this research. So preliminary results in ecology. Here we use for the current description and drifter that we built, and we deploy in different sites of the Nocivora seaweed that we see here. I don't know if you see my mouse. So we take different points, and we deploy the drifters to see how the current goes in the different farms, in different three localization of farms: north, center, and south. So here are the results. Each line represents a drifter trajectory, and now we can see and we can we prove more or less the current direction that help us for to determine the position of coral nubing stables and other kind of question that we do in the project. Here are some results about the physico-chemical parameters. Here is a dissolved oxygen that is one of the most interesting parameter because we can do the activity, the biological activity of the Capaficus, of the algae. And we see that in the middle of day are the peak of the dissolved oxygen because the photosynthesis are the stronger activity of the photosynthesis of the algae. Here is one application of geomatic from drone surveys, and I try to determine the occupation, the surface occupation of reef lagoon in different farms. So we can calculate the surface of the farms in the total surface of the reef lagoon. And here is our sampling plan for the coral nubin tables that we do it in the near of the farms and near for the reef to have the two study case and compare with the activity of the algae and without the activity of algae. So the underwater protocols, visual protocols, we use a Global Coral Reef Monitoring Network protocol to describe the benthic communities. So here maybe it's a little bit so much information, but I want just to say that we have some elements of results thanks to the great work that doing the master student from Madagascar. So I resume some data in this table, and mainly as we see mainly in the zone that we deploy this protocol, we see that the mineral component is mainly represent. We have some present of corals and macroalgae, and in some sites seagrass are very important, more in the south. So it's a current analysis. So I give some preliminary results, but I don't have so many details. Even though here I present some visualization of data. So as I say, mineral component is mainly represented by sand, and we have some rubbles and also beach rock that represent the marine surface, benthic surface. And we have also the representation of seagrass that are very interesting also to study this dynamic between the seaweed farming and the seagrass. And we are currently in the biodiversity census, but I can say now that we have in this seaweed farming exploitation more than 4 or 5 seagrass species, 2 or 3 coral genres, and we have typical reef and seagrass fishes, many oceans, and many macroalgae also are present in the different sites. So now I actually in the field for 2 months, and yes. Yes.
Sorry, you have you are over time, so I don't know how many slides you have left.
No, just 2 slides and I can go quickly. So I was just finished to say that I'm currently in the field work, and we will do it. We will complement the study because we have a lot of work to do. And I just want to show some sampling here. It is the multispectral survey by drone. Here is the our sampling plan for physico-chemical data. And just for today, it was a great day in the field, and I saw some very nice organisms. So I think we have a great biodiversity in this exploitation seaweed farming. So we need to try to preserve that, and we work on to have the information to support the society and all the partners to do this activity in sustainable way. So as I say, anthropology and environmental law, if there are some questions, and I just want to thank all the collaborators. We have a great team that work on the project. So thank you so much. If you have any question, they are very welcome.
Thank you so much, Isabelle. I think it's a very exciting work what you're doing, having a coral reef cultivation near seaweed farm. It's not very common. It has been done, I think, in Japan, but yeah, very exciting project for Madagascar. So with that, we will open the floor to some questions. I know it's a big group and it's not always easy to take the lead for questions at this time, but we'll take this time before having a small break.
In fact, there were several questions within the chat. So if some of you presenters have identified some specific questions which were addressed to you, please, if you can have a quick answer for some of them, you are pleased to start for the discussion. Yeah. There were several questions about the methodology, and some of them probably were answered. There were some questions about especially the importance of sampling sites in relation to the hydrodynamism and the direction of the currents. So as we have seen in the last presentation, there were some means, of course, to evaluate the different currents. I know that in some studies we will hear about some current methods also. So that's of course, if Sophie or others want to answer to such questions in terms of designing the sampling for in their sites, so please do it.
I can maybe say something to it. And I know that Ud is in the call, so she might also complement my answer. So in terms of the similarity of the two sites that we assessed, they're quite similar in the sense that they're really in the same fjord, and it's just they're just a little bit, there's a distance in between, I don't know how many meters or kilometers, but so the difference mainly is the exposure. But Ud, if you can come in here.
Yes, hi everyone, we're back from Ocean Rainforest. Yeah, I think you already answered the question, but can you just, sorry,
is there a difference between the sites in terms of the composition, the chemical composition of the water close to the...
Yeah. We have a much higher water light penetration in the offshore side, so we see growth further down on our lines, so in the more exposed side compared to the moderate or sheltered side. And higher waves, much higher waves in the exposed side. Current is not so extreme and similar at both sides. Temperature the same. Yeah. Anything else?
I think that was the question, yeah.
There is no more question. Maybe I will leave the Carrie Byron. I know you are based in Maine and we don't have presentation from the US East Coast today. So maybe if you can introduce quickly in a few minutes the report you've been working on for WWF. I think it's a valuable report that we are looking forward to read.
I thank you for the introduction. So I'm based in Maine on the East Coast of the United States, and my perspective is really based in the Gulf of Maine. We have done some research here trying to quantify biodiversity on our seaweed farms. We're primarily growing sugar kelp, Saccharina latissima, and our farms are quite small scale, to be able to be managed by a family-owned fishing vessel, like a lobster boat. Lobstering's one of our primary industries here. And the other really interesting thing is that our seaweed industry complements our lobster industry. So when, of course, the seaweed's growing in the winter seasons, and then we actually harvest the entire farm and remove almost all of that gear to make space for the lobstermen to put down their lobster pots and fishing gear through the summer months. So our farms are quite ephemeral and seasonal in that way, and also very small scale. And our examination of biodiversity, using both cameras and eDNA analysis, showed that there was really no impact on biodiversity or no difference in biodiversity in our farm sites versus nearby reference site. So it'd have the same benthic structure, it just wouldn't have any farm gear in it. That paper is published, Emily Scott et al. I'm happy to forward that to everybody. What Melanie was just referencing here, WWF, World Wildlife Fund, did ask me to write a report summarizing our collective and current state of knowledge on biodiversity in seaweed farms, or how seaweed farms impact biodiversity. Unfortunately, it's a bit of a tease because this is a private report contracted for WWF, so it's not going to be publicly or academically available. Paul Dobbins is the contact person there, so I would reach out to him directly if you have interest in this report. And, you know, I basically summarized the information that I could find in the literature and through a few conversations such as this about what's happening right now in the moment that might not yet be in the literature. So yeah, this workshop was very well timed to be able to give me a little bit more of a heads up and knowledge on what might be happening in terms of quantifying biodiversity on seaweed farms that is not yet captured in the literature. Thank you. I'll put my email in the chat if anybody has any direct questions for me.
Yeah, thank you so much, Carrie, for this explanation, and apologize for pointing the WWF report. I know also Sophie Corrigan with Juliette Brody at NHM, they are willing to work on studying how seaweed farming can have positive impact on marine protected area. So I don't know if it's still the beginning of the process, Sophie, but if you want to say a few words about this, unless someone wants to react to Carrie's speech, yeah.
Yeah, in case if anyone does want to speak to Carrie, just jump in. Yeah, I'm going to give a talk later about my work on biodiversity monitoring at seaweed farms. But that was like my PhD, and now I've moved on to a postdoc on the Global Seaweed Superstar project at the Natural History Museum. And something I'm quite interested in looking at is whether seaweed farms can act as other effective conservation measures. So by excluding more damaging activities from an area, such as fishing, like bottom trawling, etc. If their seaweed farms are not having a negative impact on the benthic environment, can they have a positive impact by, excluding more damaging industries? So I'm not going to talk about this today in my talk, but it's something that we're looking at working on, and we've seen the same in things like offshore mussel farms with really positive results in areas that have been degraded previously. So yeah, if anyone else is working on something like that, it would be great to connect on that front. But if not, I'll save the rest of my reflections for my talk later. Thanks.
Thank you.
I was going to say, sorry, Sophie, like my talk to you as I'm a stage, because you see in the presentation something that we saw in Ventry, the exclusion of trolling from seagrass areas. So yeah, that's something that you see later on. You can talk. Yeah.
Yeah, that would be great. It'd be great to know if anyone else is working on something like this.
Yeah.
Melanie, I actually have a question that I was asked in the chat.
Yeah.
Can I throw it back at the audience and get their input? The question was, is eDNA actually a good tool to quantify biodiversity? Because I was told it's not going to be so solid if we want to quantify some fish or some crustaceans or so on, because it's actually, they don't shed so much DNA and they just swim by quite quick. But a couple of people have asked us and said it actually works quite well. What does everyone think? Is this a very good tool to quantify really how many species there are, to say something solid about that?
Maybe Naturmetrics, I see you are in the room, and you've been a pioneer in doing eDNA as a company. Maybe you want to react to this question?
Yeah,
No?
sure. Hi, everybody. Turn my camera on here. My name is Nathan Giraldi. I have an academic background, but yeah, I've worked in Naturmetrics for the past few years. I mean, like, I think it's most important to say that eDNA is not for everything. Obviously, it can't do abundance right off without clear validation. And, you know, there's still specific things you have to be careful with. But with that said, I mean, I think especially for fish, where the sequence database is really good, I think it is a really good tool. There's plenty of studies out there showing that. I also think the benefits comes with, you know, a single water sample. You can look at pretty much the whole tree of life. But as you get smaller, you know, into the diatoms and even in the bacteria, you're not going to get the taxonomic resolution you'll get with fish. So I think, yeah, I think it can be used, but just keep in mind that, you know, it's not perfect. And I think it should be used along when you're looking at, you know, ecosystem condition and uplift and things like that, that you should also try to use some other traditional methods, I think, for a good whole picture. But for sure, I think it fills an important piece when we're looking at to assess overall ecosystem condition and uplift and changes in biodiversity.
Thanks.
Yes, for that. So I think with that we can do a quick break and go back. So in Europe it's going to be 3:30 when we're back. So in 8-minute time. So you can break, and we'll start with the presentation of Jose Maria that has been studying farm in the UK and impact on seagrass of seaweed farming.
In Ireland.
Sorry. We are back here in Roscoff.
Back here in Galway.
Okay, you are here.
Yeah.
Maybe let's wait another minute so that everyone back.
I'm trying to see if I share.
Yeah,
Yeah,
you
yeah.
can start to share.
Yeah. Like, I have too many screens here.
Yeah.
1 second. Exit full screen now. I'll put you over here. Now, share.
Yes, it's working well.
Yeah, okay. Do you see the Zoom menu on when I share the screen?
No,
Okay, it's only me that I see in it. That's grand.
I think you can start now. Now we can see your.
I
Yeah.
just move that out of the way.
Yeah.
Okay.
So I think you can start, Jose. We still have 75 people online, so.
Then you're recording, no?
Yes.
Yeah. Okay. Well, thanks everybody, and thank you, Philippe, and thank you, Melanie, for inviting me to this workshop. I think it's a great idea. You actually have a gathering of minds because there's a lot of people doing a lot of stuff at the same time. It's just for to harmonize perhaps approaches. I think that's a good thing. Yeah, I'm mostly going to be talking about the work I did for Genial. Yeah, my name is Jose Maria Fariñas. I'm now based in the Atlantic Technological University in Galway, in the west coast of Ireland. So the work that I'm doing is mostly around ecosystem engineers. So I'm very interested, always been on biogenic habitats and keystone species, low trophic habitat formers, mostly oysters and mussels, but then also as well seaweed and seagrass. So looking at restoration from that angle, and I suppose that my participation in the Genial project came from that perspective, because we're looking at the impact of seaweed farming on benthic ecosystems, as well as the enhancing of biodiversity. So I have a bunch of really great students working on everything from like native oysters to horse mussels, as well sustainable management of extraction of seaweeds and seagrass surveys, and as well you see Anthony Adugianfi there working on an INTA farm that we have as one of the test sites to continue the work that we did in Genial. So angles in terms of research on ecological interactions from seaweed aquaculture. The work for me started with the Genial project, which is in the University of Galway, and I was looking at Sophie's presentation and how much data that is collected in surveys, and that's what happened in Genial. We cover so much ground that there is a lot of perhaps need to continue looking at what we're doing there. So that work continued in ATU when I moved to ATU. So I have PhD student is Anthony Adugianfi, which is looking at the effects on benthic communities on this test site that we work in Ventry in Ireland. Looking as well at the temporal and spatial and differences between Aleria and Saccharina, because although Genial was mostly focusing on Saccharina, we had as well some lines on Aleria, and the farm that we're working as well in Cork is working with Aleria. So we have a chance as well to see the differences between the two species in terms of habitat provision. In terms of impacts, I mean, they are both kelp species. We think there's going to be much of a difference, but it's more on the differences between the two species in terms of how much biodiversity they attract and the assemblages that might colonize one versus the other. Looking at succession as well over the years, so differences between from the seeding to leaving the seaweed over the summer as well between different years. On habitat provision, so a nursery role for fish and crustaceans. So Peter Lahiff and Megan Shipton were looking at that. So master's students on what happens as well if you leave the long lines over the summer and you don't harvest the seaweed. And Anthony is working as well on the trophic impacts in the food webs. We're doing some stable isotope analysis work. So the sites that we had, I mean, Genial was in Ventry Harbour, which is the site that you have there on the top left, which is in Kerry, southwest of Ireland. And after I moved to ATU, we had a chance to work in a site that is a bit closer because the logistics of this all were a big factor as well, how much work you can do, rather than driving 4 hours down to Kerry from Galway. We have a farm now here in Connemara, which is managed by the Marine Institute. It's an IMTA system, so they have salmon farming as well as other species being grown there. Very small scale operation compared to, for example, Ventry. And the same farmer that was growing the seaweed in Ventry had another site license in Castletownbere in Cork. So we had a chance as well to go back to work with Mike Murphy. He led a sample then in his new site in County Cork. So for Genial, it was a Horizon 2020 funded project. Philippe, very well familiar with it. He was managing the whole thing, I think, to a good end. It worked well. I mean, it was 4 years. And the focus was on productivity as well, biodiscovery and increasing, like looking at phenotypes and genotypes and strain selection and all those things that, I don't know, make money in a way. But my interest was in biodiversity. There was an angle to it that it was on the impacts, positive or negative, on the ecosystem. And that's the work package that I was mostly involved with. Is that okay? Good. Yeah. So we have lots of partners in Genial. We created an e-learning course. It was one of the outputs. So the website is there. It's still active. Last time I checked. And we created an e-learning course on seaweed cultivation and monitoring protocols, which is still available. So just to compile everything that we work on in Ventry as well in the Netherlands, we have another small site in the Eastern Schelde that Wageningen was managing. So the principles always, evidence-based, gather biotic and abiotic environmental data. I work mostly from the BACI concept, before, after, control, and impact. And it might not be an impact, but it's just the presence of the seaweed farm. And the idea is always to detect spatial gradients and temporal gradients and separate these anthropogenic versus natural sources of variance, look at replicability across the geographical spectrum. Not going to go too much in detail here because I have the slides which are much nicer on the eye than just a list of like a bullet points. But we look at positive and negative effects. So the direct effect as biodiversity enhancement of the presence of the long lines, so sampling directly the seaweed, and perhaps as well the presence of invasives. So we look at that as well, whether they are an essential fish for habitat. We did dive surveys, and then later on, after we continue the work in ATU, we put some drop cameras and we did some baited cameras as well. In terms of the qualitative analysis, we have some dive surveys in terms of habitat characterization, and then some semi-quantitative analysis with subforest scores and UNIS biotope assignation, just as baseline studies before the farms go out there, and quantitative, which is photo quadrats and grab samples. So without going too much detail, that's just this schematic that we had. We look at the seafloor, and we did some grab sampling over 2 years on a gradient under the farm and away from the farm. We did as well civet transects. We put irradiance loggers just to look at this whole shading effect on primary producers, and I'll tell you in a second why. Direct sampling of the long lines, and Then as well, we put droppers just to compare the effect of the seaweed versus the ancillary structures. So the droppers had some were seeded, some were just the ropes hanging, and see what colonizes the ropes. So just compare perhaps the biodiversity effect that the farm has away from just the presence of the seaweed as just as an artificial reef. So we did a lot. I mean, the fish as well. We put some baited cameras. Like I said, we continue the work in Castletownbere, no, but we put some baited cameras in Connemara. So for the fish as well, we did some diving transects along the long lines. So the approach is all backy, and I got it informed from salmon farming monitoring. So you can see me there, yes, that's from an oyster survey, but just to get an idea, we have a 100-meter leather line, and we just put quadrats, random quadrats every 25 meters, just to get an idea of the benthos under the farm and away from the farm. So you have the schematic there. So again, the idea is that the farm is the source of impact, and then the theory, the hypothesis, as we move away, perhaps if there is an impact that becomes like attenuated with distance perpendicular as well to the current and in the direction of the current. We did that 3 times before seeding, 3 months after the seeding of the lines, a peak biomass, and then after the harvest over 2 years. So all video and photos and graph sampling as well, just to look at the infaunal communities, everything analyzed with the usual statistics, univariate and multivariate. The good thing with Ventry was that serendipitously as it was on top of this. So it is the deepest seagrass bed, I think probably in Europe, at least in the UK and Ireland, but that's 12 meters deep. So the visibility actually is quite good. There's a lot of light coming through. And that footage is from not too far from the farm, probably like 5 meters away from it. And you see all the pollock, that's all pollock juveniles. And you see the same species as well in the long lines, okay? So it was a great case study, just because you couldn't plan it even if you wanted, probably in terms of permits, to have to look at the effect of the seaweed farm on top of a seagrass bed. So you see photos here, they are under the farm and away from it. And the idea was as well to see whether benthic primary producers are affected by the overheard farm. And just to go, not to go too much into detail, what we found that it wasn't a significant effect. And in fact, a lot in some directions away from the farm, there was more seagrass in terms of density and coverage close to the farm or under it than away from it. There's some elements in terms of depth as well, that's why the seagrass become less dense as we move away from it. But there's another element as well of exclusion, I think, from pots. So there's lobster potting and crab potting and trawling in the bay as well. So what we saw again is that there wasn't much of a decline or a direct effect of seagrass on the seagrass from the seaweed farm, and as well we do some irradiance and turbidity measurements. And funnily enough, the shading wasn't really there. In fact, there was more light getting through from the farm, directly under the farm, than in the controls away from it. So in terms of irradiance, actually the opposite was true. There was more light getting through to the seagrass. And then as well, you have this sediment deposit, sediment deposition. We have these sediment capture traps, sediment traps. We saw more significantly higher sediment deposition away from the farm than under the seaweed farm. And the thinking is that it's probably acting as a sediment trap. I'm not going to go too much detail because this is all biodiversity. We dug out some papers, historical papers, and actually the seagrass in Ventry Harbor was covering the whole harbor. Funnily enough, now is just limited to the southern part, which happens to be where this seaweed farm has been for about probably 13, 14 years. I have to say as well that Ventry is used as an anchoring harbor because it's sheltered from storms. Even the Irish Navy uses it, and there's a lot of trawling and potting in the area. So, I mean, there might be other reasons why the seagrass bed is limited to the southern part of Ventry, but I think an element to it has to be as well the effect, the exclusion effect from the farm. In terms of the benthos, that's the work that Antonia Gianfi is doing, similar to Sophie and Algolesco. I think it was Algolesco you were showing your diversity data. In terms of Shannon, is pretty high. I mean, I think there is an effect as well of the fact that you have a seagrass bed there compared to controls, which is distant controls, which is mostly a sandy area. And there is no real effect in the gradient. The biodiversity is as high, if not higher, on the edge and under the farm. And in terms of microinvertebrate abundance, not much of a difference there. So we don't really see as well, even in community composition, differences. It's mostly driven by season. So you have in the summer higher diversity and higher abundance, mostly abundance. And in the winter, a decline, which follows the natural trends in the ecosystem. And the controls, probably they're not great controls because it's not a seagrass bed control, but you can take the 100 meters away from the farm as a seagrass control to see that there is no differences. The controls are all sandy areas, like 200, 300 meters away from this farm. So mostly driven by seagrass rather than an impact. I have a lot here. I try to go fast on these other results on the biotic communities in the cultivated. How much time do I have, Melanie? 5? Or is it time already?
3 minutes?
3 minutes, Jesus, okay. Right, we did a lot of sampling in Ventry and we've continued the sampling as well in Cork and in Connemara. 25 centimeters of long lines, we did it seasonal as well to see the colonization from March to July. And we left the seaweed grow over the summer as well to see what happens if you don't harvest it. So we have 2 sampling points in May and in August in Connemara and Castletown. So in brief, the succession is there. I mean, early colonization, lower diversity of species, and then it just goes higher as you get to June and July at peak biomass. Across all sites, 143 taxa, and per site between 48 and 57, which is not too different from natural kelp beds, although the assemblage is a bit different. In terms of differences between species, we saw perhaps more diversity and more abundance in Saccharina, probably because of the different structure of these rugosities and the physiology of the blade. Not much differences, but a bit higher in all sites. The difference was in Lehana Pool, where we saw a switch between Alaria and Saccharina. That's Megan's and Anthony's work, with more diversity in Alaria. And we were wondering why that was. So I'll tell you in a wee second. In terms of differences, you have most of the differences are between sites. Okay, so in one site here down in Castletown, there were more amphipods, and another site here we had in Connemara more gastropods and grazers and bivalves. So there's site-specific differences, but within each site, not much of a difference as well between the species. Other that for this reason that I'm going to tell you in a second, we realized that the ropes were different. So they were growing in the Marine Institute, they were using these polypropylene ropes that don't attract much in terms of diversity other than the seaweed itself. So when the seaweed is removed, it's pretty clean. There's no sediment in the crevices. But for Alaria, which in other locations with the same ropes usually had less diversity, it was all biofouled. There was a lot of sediment in there, and the ropes were more were Hessian, with natural fibers. So it was absorbing a lot more water, and there was a lot of sediment, and that's where all these bivalves and polychaetes. So there was a cumulative effect from having a different, like perhaps more bio-friendly material in terms of promoting diversity. So I leave it there for you. The other change, I probably are with time doing really bad. The whole thing moved onto a mussel habitat. So in both sides, we left the ropes there, particularly in Ventry and in Castletown, and it becomes a mussel reef, okay? Which is a good thing because what we saw is a nursery effect for crustaceans. So we have a lot more diversity compared with the seaweed, or perhaps a cumulative effect because the holdfast was still there between the seaweed and the mussels, with more polychaetes and more bivalves, and particularly more crustaceans. And these species are commercially important, this brown crab and and velvet swimming crab. So that was the droppers experiment that I did inventory, and I didn't have time to look at all these. I kept all the samples, and I have master's students to look at them. That was Peter Lahif doing this work, and that's exactly what he saw. So we have droppers there without seaweed, and others with seaweed. And the difference is in 3 months is that, you know, with seaweed you have more abundance, but without seaweed you still have stuff growing on the rope. So there's an effect on the structures in terms of promoting diversity. The species were a bit different, but the difference was if you left the ropes over the summer, that you have more diversity, less abundance, because you have lots of amphipods with the seaweed, which the fish are feeding on. So the shift is less abundance, but more complex communities with the mussels and all these crabs and, you know, all these crab species. And the fish surveys is something that we continue on. We have weighted remote cameras, inventory and in Connemara, snorkeling and diving along the long lines. We have shrimp pots as well, hook and line. So the design, Sophie, you probably see that, you know, similar is your design from your paper. We replicated that cross thing to see how it worked. The other design underneath is just for the bottom. Inventory is just a pyramid with GoPros. And just to show you what we've seen, lots of juveniles, inventory, and mostly inventory and Castletown bear. And all these juveniles are pollock as well, 15-spined stickleback, wrasse, lumpfish, and some surprising species here, salmon. So it's a wild salmon, so it's not an escapee from a farm. The squid, I didn't see that. It was Anthony, my PhD student, that actually picked that up. When I was looking at the footage, I missed that completely. There was a squid, rocklin, and this beautiful, you know, thornback rays in the seagrass bed, and bullheads and cat sharks. So yeah, I mean, the jury's out in terms of which species are associated with the seaweed farm and which are coming to the baited cameras. I accept that, but the main thing is this juvenile. So in conclusion, no negative impacts from all these studies, and we continue as much as funding allow us, if we can. The changes are mostly with time, your natural changes that you see in the control communities. There's commonalities in terms of what's attracted to the farm between different sites, but there is site-specific. There's lots of amphipods and lots of blue mussels at the beginning. And there's a succession that's interesting between a seaweed and a mussel-dominated community. The other angle is that they are de facto protected areas, and the Ventry example with the facilitation of the seagrass bed, I think is quite important, and I hope to get that published. There's a report on this, it's all in the Genial website, okay? So yeah, you want more info on this, I'm happy to work in projects and collaborate with anyone that wants to continue with this work, because at the moment we're probably going to end up because Anthony's PhD comes to an end. So yeah, that's it. Sorry about taking so long. I just wanted to tell you all about all this, so much data. Yeah, thank you.
Sure, and I think you kept us really interested with your beautiful pictures.
That's the thing. Like, I mean, take pictures, because that's really what's showing.
Yeah.
Okay.
Okay, so we'll go fast to the next presentation, which is from Denmark. Annette, if you want to jump in with showing your slides.
Jump in and share my screen, and I hope it will work. I'll go, yeah, so I'll just change here. So, does it work for you?
Perfect. Yeah.
That's good. Okay. So that was a really nice presentation from Galway there. Jose, really. So this one, what I'm going to present is a smaller study that we did some years ago in 2 Danish farms, where we used eDNA sampling to see if we could, look into biodiversity. And we based it on some studies that one of our colleagues made on boulder reefs, where he looked into the biodiversity using eDNA, looking into upstreams on the boulder reefs and downstream of the reef, and he saw that the eDNA would add substantially to the diver-based observations of benthic species composition in fauna and fish. But there were some caveats as well. It was really difficult to see the local imprint of the boulder reef, as it was relatively weak in the eDNA. And especially there was a miss out of many of the macroalgae species using eDNA. And he highlights that we need very much to look into the control of water masses to make sure that we're sampling in the water mass that actually passes what we want to look at. So we measure all the follow-up parameters as well: temperature, salinity, current speed, and direction, in order to be able to characterize and discriminate between the water masses and get the right signal that we want. So we try to follow that, and instead of looking into a boulder reef, we looked at 2 seaweed farms. So we wanted to use this to test if we could see an impact on biodiversity of kelp cultivation, both in the pelagic, sampling the surface waters upstreams, in and downstreams of the seaweed farms, but also looking at benthic samples, trying to compare a station right underneath the seaweed farm to a station, a reference station selected to be similar and relevant as a reference station in the same area. So the locations that we looked into here in a joint campaign was one in Grenaa. So that's a map of Denmark that you see here. I see if I can just get a little that. So you see Denmark here. So the first site was in Grenaa, where we have a small seaweed farm, few long lines, sugar kelp, only few hectares, and that is owned and run by Aarhus University. And the second, and here we had the option to do a baseline survey before the seaweed was put into the area to inform the batchy design that Jose also talked about. In Hjarnø, we had the second seaweed farm, and that's a large seaweed farm. As you can see here, it's a commercial one, 100 hectares, out of which 33 hectares were in full use. So you can see here's the seaweed farm here, and you can see it's in like 9 to 11 meters of water. And you can see it here with indication of where are the lines that are in use at the time we did the sampling. And you can see that out here, upstream, we have the reference site selected for this area. But here, this farm had been in use for commercial seaweed cultivation for years before we did this survey. So here we had no comparable baseline survey, so we were not able to do any backy design here. So what we did was that we did sampling at the time of maximal biomass in May, just before the harvest, and we used the Aarhus University research vessel for this. So we did the pelagic eDNA sampling upstreams in the seaweed and downstreams at both sites, filtering the water samples through Sterivex filters. And then for the benthics, we did below the seaweed farm and at the reference station at both sites. We used HAPS corer to get some sediment samples, and then we took the samples from those. And we froze the samples, we did DNA extraction, then PCR with eukaryote primers, and then we did sequencing and then bioinformatics afterwards. So to the results, in Grenaa that you have here, we saw that we did not have any significant difference between the reference site and the seaweed. So that's 2 different, here's the Shannon and another biodiversity index, the alpha diversity measure. So no imprint here, no difference between the reference and the seaweed site. Then again, we wouldn't expect that because it's a really very small seaweed farm we had here. It had only been 2 years in use when we did the sampling. So also in time and space, it was very, very small. In Hjarnø, we saw a different picture, because here in Hjarnø at the big seaweed farm, we saw a significantly higher biodiversity in the sediments below the seaweed than we did at the reference station. This, again, we did not have any backy design to follow up on this, and we didn't have any before the 2 sites. So we did not actually have any means of saying that this was because of the seaweed farm. We could just tell the difference between these 2 areas, and potentially this was due to the seaweed, but it could also have been there before the seaweed was employed in the area. So inconclusive, unfortunately, but interesting. Then when we look at the pelagic results, we saw that in Grenaa, again, no significant difference between the seaweed farm and the upstream. We didn't have a downstream on this one, but here you see the upstream and then coming down to the seaweed. So there's a bigger variation in the seaweed farm, center of the seaweed farm, compared to the upstream, but no significant difference. Again, small seaweed farm, short term in use or short time in use. Then for Hjarnø, we did actually see an indication of a higher biodiversity downstream than in the center. And this is the downstream here. And here's the center and the upstream. So we did see an indication of a higher biodiversity here. And you see the current direction here coming from the upstream, then to the center of the seaweed farm, and then to the downstream. So an indication, higher diversity downstream. Then again, the results are not significant. Yeah. Yes. And if we look into the results in a different way, so before we looked at the biodiversity indexes, now we kind of compare what species are there. So we look into the top 10 phylum here. So we can see that we have a lot of microalgae. These are, yeah, dominating all of the signals here. Then still we can see an indication that there are more arthropods in the center of the seaweed farm, both in Grenaa and potentially also in Hjarnø. And that would make good sense because we would expect to find more arthropods in connection with the seaweed, but also with the structures of the seaweed. Then we also see in Grenaa especially a small cnidarian signal here that we do not re-find in the Hjarnø, but cnidarians would also make sense because that would also be a potential sessile species on the seaweed and on the structures as well. But in contrast to what we hoped, we did not see any macroalgae signals at all, but that could be due to the primers used or the database that we used to identify the species. So the discussion for this, yeah, we have not fully investigated the data yet, and we are very happy to collaborate with people who are working on this in order to get full exploitation of what is in the data. We know that these seaweed farms that we have in Denmark have a relatively low stocking density. At these sites, we had less than 1 ton fresh weight per hectare at the site at the time. Whereas if you look to China, for instance, they have 151 tons per hectare at some of the big, very dense farms. So we would expect maybe a smaller signal in any case. But we really, what we will do in the future is to try and carry out more detailed studies at different sites, different times, different scales. We will need to employ backy design, so we are in collaboration with all new farmers in Denmark, hoping to be able to perform baseline studies before they implement the farms. We need a discussion on how to choose the good reference sites, because that is of utmost importance when we want to have good results, being able to be conclusive about the real impact of the seaweed site. We hope to couple with models that can estimate impact on specific site by using hydrography and biology in the modeling as well for the water masses. And then we would very much like to cooperate on optimizing and aligning species identification and the use of the bioinformatics tools, because we have a lot of data here, and we could help each other out optimizing methods to avoid collecting a lot of data that does not give us a lot of information, and just gives us a lot of work. And with that, I would like to thank all the people that participated in this study and all the projects that this was a combination of efforts between. Thank you.
Thank you, Annette, and very interesting to hear about those results, the sediments, and what you have observed at a microscopic level. I think it's very relevant to us, especially in Algolesco, so we'll definitely be in touch to understand a bit more about your protocol, and maybe we can help with bioinformatics in Roscoe. Okay, then we are waiting to hear from Sophie, and lastly from Colin Bates, based in Canada. So Sophie, I have your presentation, unless you want to share it, because Colin will be sharing.
I can share mine for ease, if that's better. Yeah, great. Has that worked?
Yeah, perfect.
Okay. Perfect. So yes, I'm Sophie Corrigan. I'm now a postdoc at the Natural History Museum working on the Global Seaweed Superstar project. But today I'm going to be talking about some of the work I did as part of my PhD at the University of Exeter and the Marine Biological Association, and kind of reflecting on the biodiversity surveys I did in light of the discussion today, and talking about, how are we going to standardize monitoring going forward in the future. So I think it's important to go back to why we're here today and what the importance of monitoring is. As we know, seaweed farming is expanding globally into new areas like Europe and North America, and South America, and all over the place. And many seaweed farms are promising biodiversity benefits, but we don't really have a solid evidence base. It's great that all these surveys are being done at the moment, but on a site-specific basis, we don't have evidence to draw upon for this. And because of this, we see that many seaweed farmers are facing opposition from people like marine licenses and local communities, particularly in the part of the world where I was working in the southwest UK. I saw a lot of headlines like this earlier in the year about anger over seaweed farms because they hadn't done these public consultations and they didn't have the evidence to show what the impacts would be. So thank you to the Global Seaweed Coalition for organising today. I think it's really important that we look at how we monitor the ecological impacts of seaweed farms in a meaningful way. And this is something I'm going to come back to at the end of my talk, but it's good to see that a lot of people have been reflecting on this and the difference between are we trying to monitor ecological impact assessments, or are we trying to determine biodiversity net gain benefits of seaweed farms? For example, is it better for farms to have no impact on the environment, or should they be trying to improve it and offer these biodiversity ecosystem services? I think it's something to think about. So Melanie has asked me today to focus on looking at how to quantify habitat provisioning at seaweed farms based on a study I did at the start of my PhD. But across the 5 years I was working on this, I also looked at how farms impact nutrient cycles in plankton communities and benthic species, and whether seaweed farms provide habitat value for epibionts, the species that live on the seaweed, and fish species as well. So I'll touch on a few reflections from this data. But to go back to this initial review paper I wrote in 2021, it was published in 2022, looking at whether seaweed farms provide habitat. It's great to see so many studies have been done since then. And yeah, this really needs revising based on this new evidence we have, which is really exciting. But this review looked at whether seaweed farms provide habitat for each of these different organism groups, so ranging from microorganisms up to megafauna. And importantly, how these have been monitored previously at seaweed farms, which techniques have been used, which survey methods have been used, and this was within the idea of whether we can standardize monitoring protocols to develop best practices amongst biodiversity surveys. So as we have seen, there are multiple methods to monitor these different taxonomic groups. And in this paper, there's a huge table which lists all of them and how they're relevant to each of these different species groups, what the pros and cons are for using each of these methods, and which surveys had currently been done. So linking to the literature where they have listed them as an example. So as well as looking at the different monitoring techniques, this paper really reflected on the best ways to monitor biodiversity at seaweed farms, and had quite a few key take-home points, which I'll try and summarize quickly. As we know, we need multiple methods to accurately monitor biodiversity. We've had some talks about eDNA today and whether that's kind of the silver bullet for doing so. Or if we're what we mean by biodiversity monitoring, are we trying to monitor everything? Are we trying to monitor particular species groups that we're really interested in? And what kind of data we're looking to get from this? Are we looking at species richness, species abundance? What kind of biodiversity do we mean? I think a lot of terms get floated around out there about this, and whether we can use a standardized kind of definition to encompass this. And just as important as the monitoring methods used, I think, is the importance of survey design. And we've talked about this quite a bit today, the importance of establishing reference sites. If you're using kind of a gold standard, like a BACI survey that Jose was talking about, where you have this before and after control impact. I think also what's really important to consider is the timing of the surveys when they were conducted. If they're conducted just when there's peak biomass at the seaweed farm, does this reflect, the whole growing season or the whole year of seaweed cultivation, or 6 months of seaweed cultivation? And how this relates to different species life histories, such as breeding and spawning events. You know, if you conducted a survey a week later, would you have still captured the same biodiversity, and whether this is reflecting on that. And going on to looking at the differences between attraction of species to a farm and recruitment, and whether we can design surveys to test for this. So looking at whether seaweed farms are just aggregating species that are visiting from nearby habitats, or if they're actually adding to the biodiversity in the coastal region, if they're increasing the number of species by providing breeding grounds. And that's something also to consider if you're looking at, you know, the age classes of species. We talked a bit about juveniles, and if that's something that your biodiversity surveys could capture as well. I think that's a really important question to answer. In terms of creating standardized monitoring protocols across farms, I think we really need to consider the differences in farms globally. So not just, you know, the difference between a tropical off-bottom eukematoid farm like this one at the top, or a temperate kelp farm like the one at the bottom, but also talking about site-specific differences, environmental differences, and the scale of the farms as well. Obviously, the farms that we're dealing with today have been quite small scale compared to, you know, the large farms we see in China, and whether we can make methods that could be scaled up at these as well. And I think it's clear that we need more data from as many farms as possible, and in order to provide these answers to policymakers and people who are interested in things like biodiversity credits, we need to collect this in a standardized and coordinated way, so that, you know, we can compare my farm sites to farm sites all across the world and see, you know, if these results are similar, or if they're really just site specific. And at the end of this paper, I also reflected on whether it's possible to integrate this monitoring into management protocols and policy to work with farmers. You know, are we just always going to have to contract scientists or consultants to do this work for us, or can we get farmers to do it as part of their standard operating procedures? So that's most of the talk, but I thought I'd give a few reflections on the biodiversity surveys I did, looking at the differences in nutrient cycles and plankton communities caused by farms, what the habitat value is for epibionts, and for fish species and benthic species as well. So if you're interested in any of these in more detail, I published a few papers looking at these and the impacts they have in relation to different things like timing of harvesting, how wild kelps compare to farmed kelps, reach out to me and ask me about these, or please read them. But I thought I'd just give some summaries to the key findings and how I've been reflecting on these since doing these surveys. We found that seaweed farms typically provided only a temporary habitat. Our farms were only seeded for about 6 months of the year, and biomass was only really at its maximum for the last 2 of those. The farmers also tended to harvest the seaweed before biodiversity peaked. So kind of thinking about what is meaningful biodiversity, what happens to all these species that are attracted to the farm, and then, you know, can we remove the habitat? Are they left high and dry, or are they then redistributed into other habitats locally if they exist, like a kelp forest or a seagrass bed? You know, are we doing more harm than good really by, you know, creating this nice habitat and then removing it? I also tended to find that good biodiversity value was actually bad for farmers that I worked with. They wanted to, you know, sell their crop, and they wanted best crop quality. They didn't really want all this fouled biomass that had loads of, you know, really exciting biodiversity for me to look at. Is this something that's really feasible? Should this be something that we're trying to quantify with things like biodiversity credits? Is it meaningful in the long term, I think? We looked at ways to potentially extend habitat value through co-cultivation with mussels on partial harvesting. But again, I think this goes back to, is this beneficial? You know, what is the aim of the seaweed farm here? Is it to provide biomass, or is it to provide biodiversity benefits? And we found that our farms, seaweeds, didn't provide equivocal habitat to natural kelp populations. They supported different species and different abundances. So by, creating this additional biomass in this area? Are we actually altering ecosystem dynamics by, introducing a new habitat that's not, equivocal to what we'd want it to be as a kelp forest, which are heralded for being really important for biodiversity? So I think all of these thoughts, I'm sorry, it's been a bit of a ramble, but are kind of concluded by this last sentence about whether I think we need to consider a seaweed farms trying to have no environmental impact, which I think is actually a benefit of seaweed farms, or are we trying to have a positive one by increasing biodiversity? And how do we do this in a way that is actually meaningful? So hopefully maybe we can open this up in the discussion later, or if you have any thoughts, please put them in the chat. On that, I'd like to say thank you to everyone who made this work possible. I had a great time doing these surveys, and I'm really excited to see all this work continuing in this community. Thanks.
Thank you so much, Sophie, and very interesting thoughts indeed. I've seen that in the chat, and it raised a lot of questions to be tackled, a lot of research questions as well. I'm not sure we'll have time for discussion, but we have one last presentation from Colin Bates from Cascadia. So Colin, I'll let you start, and then we'll see where we end up with the time.
Okay. Can you hear me?
Yes.
You can see my screen?
Yeah, but we are in the diapo mode, so we can see the next slide also.
Hey, let me rejig that. 1 second, please. Stop. Is that better?
Yeah, it's perfect.
Okay, wonderful. Thank you, everybody. And thank you for the invitation to share some thoughts with you. And for all of the previous presentations. My head is swimming with ideas and thoughts, so I hope to connect with many of you after the meeting. So the work that I'm going to share with you was done through Cascadia Seaweed, and I'm a senior research scientist with Cascadia. We're Canada's largest producer of ocean cultivated seaweed. And our products are destined for agricultural markets in the form of biostimulants. But we also have a nature-based solutions arm, of which biodiversity evaluation is a big part, and that's the context in which I'm speaking to you today. This work was done over the past 3 years, and it was a highly collaborative project. And along with the Cascadia team, we worked with 2 coastal First Nations here in British Columbia, as well as 2 labs at the University of Victoria, where the next ISS will be held. And it was funded by the British Columbia Salmon Restoration and Innovation Fund. So in British Columbia, salmon are a culturally and economically important species, but we're seeing many challenges to salmon survivorship. One of the things that's reducing salmon abundance is the loss of nearshore habitat. And here on the west coast of North America, we've seen a real loss of canopy kelp cover. And as the seaweed industry rises here in British Columbia, we wondered whether or not the coastal kelp farms that we put in place can create novel habitat for fish and other species. So that's sort of the biological question that we were trying to address, but also very germane to this meeting. We wanted to really try to understand what sorts of tools are best equipped to evaluate biodiversity effects of seaweed farms. So the work was carried out on the west coast of Vancouver Island. You'll see the map on your screen. We had 2 different study regions, each with a kelp farm. One was the Cormorant Kelp Farm in Hausat Nation territory in Clayoquot Sound near Tofino. And the second was the Burrow Point Kelp Farm in Uchuklasat Nation territory in Barkley Sound. And the kelp farms were fairly small, between 1 and 4 hectares. And we also had an associated reference site, which was as similar as possible to the kelp farm site, but unvegetated. And those were 400 to 700 meters away. So we had a total of 4 research sites, and this work was carried out from April 2022 to June 2023, so about 16 months. So we had the typical temperate kelp farm setup with ropes and floats. And we were farming Saccharina latissima. And again, our products are harvested typically in April or May. And usually our farms would be fallow for the summer months, but we kept biomass on the lines all year round. We also do have a pledge to keep 10% of our crops online for ecosystem services. So one of our internal goals was to try to evaluate how successful our retained seaweed was at providing these ecosystem services. And this is what things look like from underneath, and you can see some fish swimming around the kelp. But in discussing there tended to be a belief that if you put seaweed farms in the water, we would automatically, they would provide good quality habitat and be heavily used. But we wanted to evaluate this and throw some data at this question. So as far as what we sampled, we had a fairly robust biological sampling program. We sampled kelp parameters to quantify the amount of kelp. And then fish being our primary focus. We were interested in salmon, but we wanted to sample all of the fish. If we're down there, we might as well see what's going on. So we took 3 different approaches to sampling fish. The first is what are called fish cams, which I'll talk more about in the next slide. And these were essentially autonomous cameras that would be left out, and they would turn on every hour while they were deployed for 5 minutes. They generate a lot of data, and we weren't sure that this approach was going to be successful. So we also implemented dive surveys, 30 minutes per month, and ROV surveys, 30 minutes per month as well. We also collected zooplankton data to try to get a sense of what might be providing prey for the fish. We also had a very robust environmental sampling program with CTDs that were continuously deployed, taking data every 20 minutes. We also did water chemistry and water motion sampling. So this is really what I'm here to talk about, which is the fish cams, or these underwater cameras. And these were a modified design. If you want to find something in the literature that describes these, you can look for Xavier Mouet's paper in the journal Hardware X from 2020. But these are cameras that are housed in PVC underwater housings. And then we made some mounting clamps so we could hang them from the farm or from dedicated moorings in reference sites. They have a camera unit, and they're based on a Raspberry Pi microcomputer. And they have a scheduler board that tells them when to turn on and off. And the bulk of these cameras is the battery packs, the yellow that you can see in the pictures there. And these can be deployed for a long duration without needing to be tended. So we would come and replace them every month, and they would just sit in the water and turn on and off and see whatever they see. Oops. So we had 12 cameras deployed for 16 months, more or less continuously, 3 per site. And then again, as I've mentioned, each camera would record 5 minutes of video. So they would just turn on at the top of the hour during daylight hours. They would also turn on at night, but there would be no light, so they wouldn't gather useful data. They had about an 80% success rate. So some cameras were lost during storms, or some cameras flooded or wouldn't turn on, but more or less quite successful in their ability to gather data. And we gathered over 16 months about 8,400 hours of footage. So this is very daunting for a human to process. So we also engaged with some academic researchers to develop computer vision algorithms that could comb through the data. And we ended up using a sort of open source model called YOLO V8, and we also developed a proprietary anomaly detection algorithm. YOLO needs to be trained to see and identify and count fish. So if there's something novel, we wanted to be able to catch that as well. So that's why we developed InReach. And you can see on your screen pictures of fish being counted. The computer will draw a box and then assign an identity or a hypothetical identity to whatever it sees. And then the number is sort of percent confidence in that identification. So I will show you a video. Hopefully this will come through. There's a school of herring, Pacific herring in the background, and the computer starts to draw boxes around what it recognizes to be unknown forage fish. And you can just see the size of this school. It's going to basically cover the whole screen, and it's very difficult to count fish schools of this size, but the computer does a good job of identifying individual fish. And as I skip it forward, you can start to see the fish a little bit more as individuals. That's a good example of the algorithm working. Here's a single fish. This is a rockfish, which when it's facing the camera is sort of hard to identify, but then it comes side on, and the computer's quite good at identifying this individual fish. Here's another school of forage fish, a little bit less dense, so you can see better again how the computer will count and identify these fish. Our primary focus again was salmon. So I'll show you a video. Here's a salmon swimming through the kelp farm. And you need about 200 solid training images to make the algorithm competent to count and identify a fish. We didn't get that many salmon, so we were only able to identify salmon to family. But with more training, this algorithm would be competent to be able to count and identify salmon to species. Oops. All right, so as far as some of the results, we had over 2.7 million fish observations, and we found that habitat use varied more so by location and season than by farm versus reference site. Again, that idea that if you put a farm in, it's immediately going to be heavily used by fish was not borne out, because we found one of the research sites had the vast majority of observations. The Burrow Point sites, we found 95% of the fish there and less than 5% of fish at the other cormorant site. We also found, surprisingly, that the reference sites had more fish than the farm sites, sometimes maybe two times more fish in the reference sites compared to the farm sites. This was largely driven by those schooling forage fish like Pacific herring, which have an inbuilt behavioral predator avoidance approach. They'll shelter under each other rather than under the farm, whereas other species, as I'll show you in a second, were sort of found more in the farms because they preferred to shelter under physical objects. We also found that there was a seasonal effect, and that the abundances of fish along the x-axis, you see the sort of month of sampling, and fish start to come on around May, and then peak in midsummer, and then by November or December are gone, more or less. And this is interesting to some of the questions that were raised earlier in the session about what happens if you remove habitat. And our typical kelp cultivation season is shown in the blue boxes in the bottom, so it's sort of December through to April or May. So the peaks of fish abundance did not coincide with the cultivation season. So, and it does suggest that if we leave kelp on the lines in the summer, that that retained kelp would be used as habitat. As far as the diversity of species that we found, there was about, I think, 20 taxa total that were seen throughout the study. So the algorithm was competent to identify a whole bunch of different species. And we did find that the biodiversity of fish was higher in the farms relative to the reference sites. So even though there was more fish in the reference sites, there was more taxa of fish in the farm sites, with the red line being the Burrow Point kelp farm, and the green line being the Burrow Point reference site. The blue line is the cormorant farm site, and the purple line is the cormorant reference site. Lastly, we found that the counts of fish and the relative use of farm habitat varied depending on what species you were looking at. So typically for the perch and salmon, we found that they were found more in the farm sites compared to the reference sites. The rockfish were found more in the farm at one study site, and there was no difference at the other study region. The forage fish, the unknown forage fish, were found more in the reference site. And again, that was what was driving the sort of more fish in the reference sites. And when it came down to the Pacific herring that got identified to species, this was no difference between farm or reference site. The tube snouts and unknown fish were either more abundant in the farm or the reference site, depending on which region you were looking at. So we found that the stationary cameras paired with AI were really effective at yielding high temporal resolution data and automating the counting. So again, we had hourly resolution of what was happening in the farms with regard to the fish communities. I'm not going to present the dive or ROV surveys, but we found that the stationary cameras provided a much richer data set. The dive results were similar to the stationary cameras, but the ROV surveys didn't really turn up much at all. There was fairly high effort to train the AI algorithm, but now that it's trained, it can be deployed quite effectively in terms of reducing the effort to apply this in different sites. This sort of work is computationally intensive, so it's not necessarily open to everybody with a laptop. But again, the collaboration is really the key in making these sorts of studies work. So as I've already mentioned, the main conclusions were that the fish cams were a really effective way of providing high resolution semi-automated biodiversity monitoring solutions with broad application. So these are not restricted to just sampling in farms. We're also looking to apply them in our kelp restoration projects and could be used in other projects involving seagrass or other habitats. The fish cams outperform the ROV and dive surveys, and those results are being written up by a graduate student, so will appear in the literature at some point. The biological results were that the kelp farm use by fish varied by region, season, and taxon group much more than the farm versus reference site. And we found that the fish primarily use the kelp farm habitat outside of the cultivation season. And the farms have more fish taxa, but fewer individuals compared to nearby reference sites. So what's next? Cascadia Seaweed is involved in commercial production of fish cams. So you can get the plans from the original publication. We've made a bunch of changes for efficiency and effectiveness and can build them for you, but you can also go out and build them yourself. And we're currently working with an AI consulting company to develop something called Seawave, which is an online video analysis platform. So if you wanted to put the cameras in yourself and then upload the data to the platform and have it analyzed using computer vision, then that is possible. And yeah, we're continuing to look for collaboration opportunities, both locally and internationally. So if you see anything that you like in this presentation, feel free to reach out, and happy to chat more with any of you. Thank you very much.
Thank you, Colin. Very, very cool tool. More than 2.7 million of fish captured in 9,000 hours of video. Very impressive. So we've run out of time. We're actually 5 minutes extra to what we committed to. So unless someone has a burning question, we'll close the workshop and publish it online. I will contact the presenter to check if you're okay for your presentation to be online, of course. So yes. If you can stop sharing your screen, Colin, we'll see all the gallery.
Thank you. Sorry about that.
And yeah, I think there is no burning question. So thank you all for your attendance. I hope you find this workshop useful. I personally find it really interesting and lots of insights shared by all of you. So thank you again for the presenters, and looking forward for new collaboration within the biodiversity field. I don't know, Philippe.
But I hope with Melanie we will be able also to sort all the comments in the chat, because that could be useful synthesis of our meeting today, and especially if we can really take the main questions which were raised, and also some of the reflection which were in some presentations. That could be interesting, because that could help you to contact other people and to tackle this equation for further in the near future. So, and hopefully, of course, we will meet during the coming year in so many meetings which will be organized, as it was mentioned by Melanie, especially at the International Seaweed Symposium. We are eager to reach Victoria, especially to discuss also directly with the colleagues from Cascadia, but also other meetings which will be organized in Europe, and especially in the context of the UN Ocean Conference. And so we will have opportunities, and I'm sure that will trigger some interaction between us, which could be beneficial to in fact, path the way to provide much more standardized approaches to which could be, of course, agreed by scientists, but which will be developed also with the professionals, the farmers, because it's important to work together, because the challenges and the concern of the farmers are very important to be taken into consideration for developing the design of survey and so on. So that's what we can at the moment say. Of course, it's a pity that we are out of time, but it was really a great workshop. Thank you so much for all the speakers for their excellent talk. And we have covered really a large series of questions, and there is still some remaining questions, which are in fact sometimes also linked to some aspect of the biodiversity which is less investigated, and especially in terms of interspecific diversity. We didn't talk at all about that. We talk a bit about the introduced species, and of course, a part of the seaweed aquaculture is nowadays developed on the transfer of species from different regions, ecoregions, and so when we are dealing with tropical seaweeds, it is an issue. When we are also dealing, for example, with the development of giant kelp in Namibia, it's an issue. So there is many questions which remain to be asked and discussed all together, and of course that will require other workshops on meetings. Okay, so thank you to all.
Bye, everyone.
Yeah.
Have a nice
Thanks.
evening.
Yeah, and thank you for putting the workshop possible today. Yeah.
Thank you.
Thank you.
Thanks, everyone.
Bye.
Goodbye.
Bye now.
Take care.